How are you today? Welcome. Welcome to the third set for the Princeton Review, the AP Bio series. And I think by now you know who I am, but anyway, in case you haven't seen the other videos, I'm William Bill Calvo, teacher tutor here at the Princeton Review. Here's my email, as you can see, and I teach a wide variety of subjects, mostly on the science and math side. Anyway, so we're here again in our journey towards AP Bio, and just remember that our goal, my goal, is to help you achieve your goals, and that's what we're going to do. Today, again, we'll have a lot of content, but there will definitely be some time to talk a little bit more about the free response kind of questions that you're going to see on the test. Okay, so let's get moving. All right, so again, some of these things, if you look at the video, you'll see a link for the other two videos shortly. But anyway, your test is on May 18th, according to the last information that we've got. There's a makeup date, and there's also, there'll be two questions, one at 25 and one at 15. Okay, you'll have some time to upload your work. And basically, we talked about this last time, 1 to 6 are the units, and you can find all that information on the College Board website. By the way, I will be looking and doing my very best to keep up with any chat questions that you have as best as possible. Here's the structure of the test. Basically, it'll be weighted at 65 to 35, and you'll have two questions. Again, you can find this information based on the College Board website in terms of getting to understand the questions. The first question will be a little bit longer. There'll be no multiple choice. Again, you'll be able to have a couple of ways to input your material, and you can find some practice prompts for the two questions here, as well as full line information, full guides, etc., that you would have everything. Same here as well. Here you'll find course description, some sample questions, some free response, because as you know, because of the situation that we're in, the test is obviously has been modified according to the College Board. I've alluded to this before, so I put this in, and I just, this time, to make sure. The material that you'll see on the test is going to be basically very long and extremely shallow. So you could say, like I've used the analogy of a ribbon, or you could say a mile long and an inch deep. The thing is, having a broad understanding of pretty much the main ideas of which they're going after is paramount here. And it's not like you have to know absolutely every single detail. There's no way that they could do that the College Board could test you on every single detail. This is technically the main ideas. And again, you find this information in the guide for the College Board about either evolution. And again, this is sort of downplayed now on this one, even though it's still there. It doesn't mean that it's one of the ideas, but they haven't modified that. Energetics, storage information, and systems. Again, the big idea about how biological systems work with energy, how they store information and retrieve it and transmit it, and basically how they interact with each other. And that's pretty much the bulk of what you'll see in terms of big ideas. Please look at the website. You'll see, or at least the Princeton Review website, you'll definitely see a link to both the videos that we've done here, etc. Basically, in the first video, we talked about chemistry of life. And these percentages have been upped a little bit because these were based on 8 units, where there'll now be 6, as you saw before. And cell structure is what we talked about basically between that, as well as energetics is what we talked about last time. So you feel free to go check out the website and take a look at those. What we'll talk about today is, again, it's probably going to be more than the 15%. Remember, you only have 2 questions. If you're looking at a percentage, it's a little bit more difficult, but chances are you'll see something similar to something related. We'll talk about the cell cycle, touch on it. Again, touch on mitosis, meiosis, some Mendelian, non-Mendelian inheritance patterns, and then finally rounded up with DNA transcription, translation, and a couple of other things. Okay, and that's the game plan. As we pointed out before, you probably won't see anything, but based on what they said, you know, not having units 7 or 8, natural selection or ecology should not be on the test based on what they're saying, because that's the unit that they're describing. All right, so like I said, we're going to talk a little bit more about content here, and then we'll try to give you some examples, or as best as possible, on how to attack them. All right, so in terms of this, obviously DNA, so you'll need to know the specifics. I could say the best way, the broad range specifics, the differences, and so forth. It's not like you'll have to draw the structure or anything like that. But basically the idea of a nucleotide, 5 carbon sugars, phosphate, nitrogenous group, and knowing what that is, and that these are the components. Okay, and you're either going to have adenine, right, AGCT, in any way, shape, or form for this. Okay, doesn't hurt to know the 2 purines and the 2 pyrimidines in terms of that. There's the phosphodiester bonds that are between the sugars and the phosphates that you'll also need to know that exist. And again, this is a lot of vocabulary too. So it's this going through all the different things in order to be able to talk about it. And then you'll see how in a free response question, you can see how this whole thing works. The gist of this, if you were just being able to describe this and remember that your stuff is written, your explanations are written, you have a 5' a 3' end with hydrogen bonds that are forming the double helix. Okay, either AT, right, or the reverse, or GC is going to be your base pairing, and they're going to be complementary to each other. Remember, you'll have a 5' and a 5' and a 3', and notice the 5' and the 3' are always opposite in terms of this on the other strand. That's the anti-parallel. Okay, hydrogen bonds are very important, at least to recognize where they are. In terms of a gene, right, we call these recipes in terms of the alphabet, if you will, and there's nothing wrong with saying that. It's basically a good analogy in terms of that. The genome's got about 20,000. That's just something that's interesting to know. And of course, putting this all together out of everything set up as a genome, and each, you can say, piece out of that of DNA is a chromosome. Okay, you've got histones that are very important, which are the proteins that things are wrapped around, and then those are bunched together into a nucleosome. All right, and the key thing is this will all have to be unwound when you start reading. So now let's take each of these processes and let's talk about the basics of what you have, and it gets summarized a little bit later. If you're going to replicate the DNA at the DNA level, okay, you should know, right? The first step is to unwind. So it's going to unwind, and it's going to need some kind of enzyme, which is an ASE at the end of it, which is a good way to remember the enzyme. Okay, and this is going to be some kind of replication fork looking setup like this, and this is a good way to be able to visualize it in your mind, and if you need to write it down in words, you'd be able to do that. Okay, so it starts out at an origin, and then you have, right, ACEs also that are going to, in this case, topoisomerases that are going to cut and rejoin, right? And they're going to prevent any kind of tangling, which is what you don't want. And these things are going to also work with DNA polymerase, right? That's going to actually do the additions and so forth. You need to know RNA primase, adding right, and associated with its RNA primer. Now, during replication, what's important is you're going to have your leading strand. You need to know the leading strand and the lagging strand and be able to describe that. Again, I can't say for sure that'll be a question, but at least, and I guess this refers back to the other thing I was saying, you could write these words down and make sure on a clean piece of paper. And again, this is just my my suggestion to fully understand something, to know that you can understand something, is if you take a clean sheet of paper, a notebook or something, and you write something down and you describe it. And if you do that accurately, then you know that you know the material. And again, that's just, like I said, that's the way that I've learned a lot of these things. Okay, and the Okazaki fragments are associated with the lagging strand. And again, there's, you know, you can take your time and make sure that you go through all the pieces. I'm just presenting the pieces here so that you could try to make, you know, it's almost like trying to refresh your memory, right? To knock out the cobwebs that you had. Okay, so you get fragments are eventually linked together with ligase. That's going to put them together, and then your setup is this. And you won't have to draw any figure like this. Remember, and we'll refer back to previous videos, you do have an open book test, and there are you know, limitations to that. You're not going to have enough time to sit there. They're not going to ask you a question that you just simply cut and paste, or something that isn't going to involve some kind of reasoning type of skill. A story about semi-conservative, because of the way DNA replicates, and it conserves half of what you're dealing with, so that's semi-conservative. And it's important to know that the very end of the thing can't be replicated, because the DNA polymerase has to have space, so it's going to bind the template DNA. Those are telomeres, and they get shorter over time. And these bullet points are important to pretty much keep in mind. Helicase is going to unwind, DNA is going to, polymerase is going to add, ligase is going to bring together your Okazaki fragments, topoisomerase is going to cut and rejoin, and RNA primase is going to catalyze. And remember, the idea of a catalyst, which we talked about, gets involved in the reaction but does not get consumed by the reaction, speeds up the rate of a reaction, and so forth. So those are good pieces of information to know. All right, the central dogma, as you know, in terms of everything, is to basically go DNA to RNA. We know it's DNA to RNA to protein. That's your central dogma, which is a really cool way to think about it. But anyway, so you got to turn it into RNA. So then you take the RNA and send it out, and it often gets turned into the protein, which is the whole goal of this thing, is to go on with life per se. You transcript and then you translate. So make sure that you get that straight. You do this in the nucleus, and you do the second stage in the cytoplasm, and that's called a dogma of biology. And again, you could, you know, that'd be something you could refer to, but it's not like you'll have a question on solely that. RNA is single. So here are some specific points that'll help you as you go through to make sure that you get all this information correct and, you know, accurate. Accuracy is very important. RNA is single, and the 5-carbon sugar is ribose instead of the deoxyribose. Okay. Notice that you have uracil when you're dealing with the RNA nitrogenous bases, and that replaces thymine. So this is a very nice summary chart. Make sure that you have a handle on it, which is the sugar that's involved between the two, DNA and RNA, and what are the bases. And again, this is just another kind of thing that you can arm yourself with if you were given data and you had to deal with some type of reasoning. As long as you have these straight, you should be all set. So it's really good to summarize that, or make sure that if you had to write this out, what are the differences between DNA and RNA, you could write them out, and then that shows that you know it. Okay, sticking with RNA, there are 3 types. There's your messenger, your ribosomal, and your transfer. And they're all based, obviously, these are the notations associated with it. Messenger is the temporary one, and what's going to be produced in the nucleolus and make up part of the ribosomes. That's easy, your ribosomal. And finally, you have transferase, which is going to shuttle, and that's your shuttle between your amino acids back and forth. And it reads the message from the mRNA, so that's where they are linked. Okay. You have 3 types when you're dealing with RNA in order to transcribe. It's got to start, it's going to elongate, and it's got to end. So these 3 things, very similar to other things you may have learned in terms of you have to have a starting phase, a continuation, if you will, and an ending phase. And in order to do that, you need certain sequences of DNA. We can call a promoter basically like a runway, right? Where the plane starts, that can't just get up and go like a helicopter. A plane has to start revving up and get like on the runway on its way to the air. Okay, so let's continue on with this, understanding the idea of how it transcribes. RNA, of course, is single-stranded, as you can see, so you only have to copy one of the strands of the two DNAs that you have. That's going to serve as your antisense. It's got all different names to it, and it helps to use that depending on if you get a question that has that. And then you have the other one that's your sense, and what you're going to have in terms of coding. Your RNA polymerase, when you have a, you could, whichever way you like to remember it, but your polymerase is going to build. It's going to add onto the 3' side, and it's going to have a starting site. Okay, starting point is where you're going to have to go, and it travels along with the and builds up the RNA so that it's going to be complementary to the DNA, of course. Now, how does the RNA get processed? Okay, well, it depends. In the prokaryotes, that's where it ends in terms of the mRNA. But in the eukaryotes, RNA is going to have to be processed a little bit before it can leave the nucleus. Okay. Exons and introns are important, and then understand the splicing, which is a spliceosome. Okay. In addition to this, you have your poly A tail on the 3' side, and you have your 5' GDP gap as well, also added to the 5' side. When you translate, okay, that's where you got to turn the mRNA into a protein. Okay, and there's a couple of things in here that are important. First of all, of course, you have to know what a codon is in terms of that, and you do have like basically a good explanation is you have to wait for a translation to initiate and for the correct sequence to arrive, and that's where the transfer comes in, because the transfer is going to have to transfer that back. Okay, so one end's going to carry, we're talking about transferase in terms of the translation, so the next part of it is actually carrying the amino acid, all right, and one end is the anticodon, and that's going to have your 3 nitrogenous bases. That's the codes that you'll have access to it, like I said, because it's an open book, but chances are they're not going to ask you anything that you would have to know in terms of that. And if you know a couple of these, I think there's a slide where you can actually just refer to. Okay, so there's a couple of things in here, but basically notice the way that it works, and you can refresh your memory in terms of how it goes about. It's basically elongation, starting out, the 3 binding sites, your A site, your P site, and your E site, and you're basically shuttling from the A to the P to the E, and basically your start codon is for the initiation of the synthesis, whichever the case might be, AUG, and so forth. If you're going to start with something like methionine, which you have access to, again, in your tables. All right, so you go A to P to E in terms of this, so that's good to refresh your memory, and you have a start, and you're going to code for methionine. Okay, then you're going to elongate, and in terms of elongating, there's pretty much not that much to know. It's just how it continues on. Everything's going to be in triplets with the codons, right? And each amino acid is going to be brought to the mRNA, and it's going to be linked via a peptide bond by the other amino acid. So bonding is always an important thing to mention, because they could ask a question where they're asking how this thing could occur. And while I'm not saying that's definitely what I'm speculating on that, understanding the bonding between things is always very useful. Okay. So basically, when all these amino acids link up together, then you have your polypeptide, and that's how you form. And that's basically the gist of it. You have a stop codon that's going to stop, and you go with this. All right, so it's good to review a little bit again. Again, I threw a lot at you, but it's cool to just lay it out and say, okay, so let's talk about this again to review. You have transcription. You're going to start mRNA being created from a gene segment of DNA, DNA to RNA to protein. Eukaryotes, you're going to have to process, right? You're going to have introns, coding or non-coding sequences, and a 5' and a 3'. All right. Now you're ready to translate. Your mRNA is going to go into a ribosome. Okay. Anything that's free-floating amino acids that are sitting around, they're going to be picked up by your transfer RNA, should say, and then shuttled over. And the mRNA is sitting there waiting and saying, thank you very much. And then you translate, use the anticodon of the tRNA, and carry the appropriate amino acid base pairs. As you match things up, then ribosome holds things in place and then allows the peptide bonds to form, because you got to have stability, and then continue on with stop codon, and you're all good to go in terms of that. In terms of gene regulation, the largest point they mention in terms of this is basically, because what you're asking yourself is, what controls gene transcription, and how does an organism just basically express only certain genes? Okay, well, that's because the regulation of gene expression, which is also something that you were exposed to, I'm sure, it can occur at all different times. So the largest point is going to be just before the transcription, and they call that pre-transcriptional regulation. And that can also occur. It can also occur post-transcriptionally, but the thing is, it's usually in the discussion on the pre-transcription. So that's going to start right where your DNA is going to be unwound. Your polymerase is going to bind at the promoter, and that usually, there's a lot more to this, but probably stuff that you don't have time and won't be asked to explain in terms of that. And there's transcription factors. They're either going to allow or inhibit, depending on the case. Okay. Sometimes, like it says, changes to the packaging is going to alter the ability of the transcription machinery to access the gene, and that's true even if you were looking at an assembly line and you saw, if you took something and put it in a box, some kind of product, you have difficulty retrieving it if it's already packaged. So there's certain changes. Also, the wraparound histone is going to make it more difficult in terms of being able to access to do any kind of changes. And again, sometimes you do see post-transcriptional work, and that is when you're going to have a cell that's going to create RNA, but for some reason decides not to. And again, to take it further. Okay. Now, embryology obviously is an important part of biology. When you start looking at the changes, the successive changes in the states of everything, that's the morphogenesis. And of course, the regulation that occurs in the embryonic environment has to do and involves the fertilization, all right? Because that's going to have the zygote go through a whole bunch of cell divisions, and you won't have to note all the different names to them, but there's all kinds of names, blastulas and all these other names that they give these. But this is an important division of biology that people study and so forth. But as these things occur, they become much more specialized, increasingly differentiated. And again, you don't need to know this at this level for this test, but you can actually, if you go on and study, you'll know where different parts of different organs, skin and so forth, where they've actually originated from what portion of the specialized embryonic development and so forth. You have genes that turn the early embryo into future this, and such, whether this is going to be the skeletal muscle or the heart or whatever the case is. And there's a subset of those called Hox genes. And everything has to happen, timing has to happen in terms of this, just so that you have all this to work on. When you have, and this is the basis of something that you'll pretty much, if I had to be a betting person, I would say that you'll have some kind of discussion or something that's going to mutate. But if you have that, then you have some kind of what's called an error. But obviously, these things sometimes can be done experimentally, right? So it's like you're creating an error. Um, but basically, a lot of mutations, and you have the basis understanding of this, that it's because the DNA is either damaged or it can't be repaired or not repaired correctly, etc. And you can have these chemicals or whatever the case might be doing this type of damage, and it's a common thing to note. A question is, what kind of environmental factor might make a bacteria inhibit transcription? Well, I mean, I can't speculate in terms of that, but definitely some kind of, one way to say this is I would talk about anything that would be, in terms of the environment, I would say something that would physically separate the bacteria from separating pretty much something from the outside environment. If you want, we could elaborate. Did you have something else in mind in terms of that? There could be a couple of different things. It could also be a chemical interaction that you could have in there. In terms of that's a very detailed question. But I could get back to that, or we could talk a little bit more about that in order to speculate. But that's one of the things that you would want to consider if you consider the environmental factors about how antibiotics are used and to go attack cell walls and so forth. So I would look at it a pharmaceutical kind of approach. So that's probably the first thing I would think of now that I'm thinking about it. But it's a good question. So a mutation is going to occur, and then what are the kind of things that you can have in terms of chemicals or radiation? Well, one of the things that can happen is obviously that this is going to occur, but the key thing here, and again, it's hard to speculate on exactly what they would ask, but if you remember that this could be a place where some kind of chemical or radiation would occur, if this was some kind of discussion as to where would you have the person's been introduced to some type of external factor, and where at what point would that be if you were describing this in terms of the origin of something. So I guess what I'm trying to say is that chemical and radiation, for example, we all know that they do damage, but they do damage at that level, and understanding, at least being able to just say that would be enough to be able to at least have a basis as to where it would, because it's all about timing. Same thing with pharmaceutical. Without getting into an elaboration, there have been drugs in the past that have created birth defects, and so a very famous one called thalidomide. But again, it's not like you'll be asked this kind of thing, but the point is that these things were discovered to create mutations and so forth, and of course people wound up with birth defects, and it was obvious in terms of that. Okay. All right, getting back to this, some things about base substitution. You have nonsense, missense, and silent different types of mutations in terms of this. And again, it all depends on what you have. And again, in terms of this. Now, this is obviously important, right? We're talking about gene arrangements when you deal with duplications, deletions, things that get inverted in translocations. Bacteria, like Christian had mentioned before, they come in many sizes. They are also pathogens, etc. Now, this is a popular one, right? And there is no way I can describe, I can even, speculate on the fact that everybody talks about corona, and that's the thing. So who knows if they decide to ask a question on that? I have no idea. I guess we'll find out. But the point is just understanding the idea of the basics again of your DNA and RNA, and the basics of a virus that has two parts, a capsid and a genetic material. That would be enough to base that. You wouldn't be getting anything, and I'll talk a little bit more about the strategies to thinking again, which is a review of what I talked about before. But you're not going to be asked some specific detail about something, especially when its current event that a lot of people still don't understand this virus kind of thing. So a good, solid, sound reasoning and sound basics on these kind of things, reviewing your vocabulary and utilizing it, of course, that's the key to addressing anything should you get a question about some other kind of virus, etc. Okay, knowing how they're very specific, they infect a host and go from there. Okay, and again, it's obviously a common thing, but there's no guarantee that they would do this, especially, you know, since there's so much knowledge, there's so many things that we don't know about it right now. So I would not get focused on it. I would just make sure I knew the basics in terms of that. Bacteriophage is also, you know, good to know in terms of a discussion of that. Now, recombinant DNA, you probably heard about this before. Again, just understanding the basics of it is important. You're just going to combine DNA from different places to create something that's going to be different and not necessarily found in nature. And you can imagine there's all kinds of things. You may go on and do research, and all this is a very common thing. And this is where you can introduce a eukaryotic gene of interest, like insulin, into a bacterium so that it can actually produce. And they're trying to do this with so many other things. So if you just have a cursory knowledge of that. If you hear about the coronavirus, they talk about, well, here's a PCR reaction. It takes a long time to do. And again, you know, it doesn't have to be corona, it could be anything else that you could say, but understanding PCR, the very basics of it and how it works to create many copies of genes within a few hours, and how this works too. So you can review that in terms of that. And it goes through the different cycles. But again, these are just part of what we call biotechnology. So there's the recombinant idea, the PCR, where you have, you know, a machine like a thermocycler, and you heat the tube and you cool it down, and then you warm it many times and so forth. And every time you heat it, the hydrogen bonds are going to break, and you're basically separating the double-stranded DNA. And then as you cool it down, those primers bind to a sequence, and they flank the region of the DNA that you want to copy. And that's what you're trying to do. So then when you warm it, the polymerase binds again. Okay. And that's basically the gist of this. Once you go through the different cycles, the double-stranded DNA segments are going to be copied into 4, and that keeps repeating so that you could do that. And that helps along with trying to figure out all kinds of other questions that people have in terms of trying to figure out different things that occur. Okay, now you also have transformation. This is the thing with insulin, is a good example of this thing. Okay, so now it's made by bacteria and so forth. So we talked about that. Okay, electrophoresis, just another technique that's used where you're basically looking at molecular weight and charge. So you could be given some information on this, and all you'd have to do is describe it. It has to do with molecular weight and charge. And because the DNA and the RNA are negatively charged, they're going to migrate towards a gel that's going to be positive in this electric field that's all set up. Okay, and with that, you've got other things. You also have with that, you also have the fragments, the restriction fragment, length polymorphisms. They call them RFLPs. You may have seen that. Or you could do the DNA fingerprinting that they talk about, where you're going to produce these polymorphisms from DNA and at a crime scene, you know. And again, you know, who knows, but that's the whole idea of the forensic things that are going on in terms of being able to look at the scene and tracing back the DNA. You compare the DNA from different people in a database, and that's how you do that. And then there's DNA sequencing here, figure out the order of the nucleotides. So in the sequencing thing, the order is very important. All right, so let's go through a summary again. DNA is your genetic. DNA's got your deoxyribose sugar, phosphate backbone. 2 strands are going to be 3' and 5'. They're going to twist. You have your double helix. Adenine's always going to be with thymine. G is going to be with C. Everything has to do with the genome, divided into chromosomes, subdivided into genes, and that's going to encode a particular recipe. Okay, so it's good to have this overall thing, and then fill in the blanks as you go on. Then we talked about replication, and that's going to have 2 copies of the DNA for division, and that's going to have your helicase, your topoisomerase, right, your primase, your DNA polymerase, and make sure you know it's RNA polymerase, right? Your DNA polymerase that's going to read, so make sure you know the functions of each one of those in case you had to describe them. You're going to replicate, and then you have a lagging strand, Okazaki fragments, which are going to be later ligated, right, joined together. Transcription is going to be building from RNA to DNA, right? And so you have to have your RNA polymerase, in this case, right, reading one of the DNA strands, add the nucleotides, right? And what else did we talk about? Except that you've got uracil in this case now. All right, so you have your messenger ribosomal and your transferase, and they're all different parts that can be created. And that's going to be processed in the eukaryotes, introns, spliced out in your 5' and 3' poly A tail that we talked about. On the translation side, you should have been exposed to the idea of the cytoplasm for the, as the mRNA is going to carry the message, right? Ribosome is where this whole thing works. mRNA is going to pass through the sites, A to P to E, right? And then you're going to read it in triples called the codons, and each one of those is going to be, well, you have a complementary, but then each one of those is going to be associated. Sometimes you have a wobble pairing, which is also something that you might have heard of, and your tRNA binds to a corresponding amino acid, and the peptide keeps growing, okay, till you get to your stop. Gene expression has to do with the transcription factors, whether it's pre-transcription mostly or post, and this is going to be dynamic, meaning it's either going to increase or decrease, okay, levels. So dynamic is very important. It's not like everything just occurs and stands still. There's always things in time as things move on. Mutations can result from the DNA or the mRNA message. Mutations can occur. All kinds of things. You can delete, you can add, you can switch when you shouldn't have, and so forth. Okay, and then we talked about different parts of technology, the recombinant PCR, bacterial as well as gel electrophoresis. Again, just knowing the basics of what you need to know should get you all set with what we're aiming to shoot for. Okay, so let's continue on with this. We've got a little bit more material. Now let's talk about cell communication in terms of the mitosis, meiosis, and the basics that occur. Now, before we get there, a review of what we talked about in the cell cycle in the last video, interphase, and then the mitosis part. Okay, so inter is everything that occurs in between with your stages, and you have the G sub 1, S, and G sub 2. Most importantly is your S phase, and that's where you're going to have your replication. Okay. We mentioned, again, your cystic chromatids, your centromere, again, just to get the vocabulary rolling, and specific checkpoints where you're going to have this. A common thing that's talked about is apoptosis, which is some people have referred to it as cell suicide, but I mean, it's basically a programmed cell death. And that's where a point where the damage is so extensive that it can't be repaired in terms of that. Okay, so you can note some of that. Again, should you ever, just look at cancer from a general standpoint, and obviously to this day, there are many questions, but that's basically when you have abnormal control of cell growth in terms of that. And oncogenes are ones that are involved in the cancer, and normally you're going to have proper growth and cell regulation to occur normally, but you're going to create some kind of other issue in the cancerous cells. So it's clearly that kind of programming change, and things that people still to this day are trying to solve. If you have a suppressor gene, that's going to produce a protein, right, that's going to prevent this conversion, and so forth. So that has to do with the CDK cycling. So in essence, to just mention this generally, and we talked about it, like I said, in the last one, plus you could do your review again. Interphase and mitosis. S phase is the important one. G1 and G2 are stages in the interphase. Checkpoints occur. Cyclin proteins and so forth are going to work together, and oncogenes are going to promote the cell growth, and suppressor genes are going to inhibit the cell growth. All right, basic understanding of the stages of mitosis. Again, the kind of thing that you need to understand in each one of these. So please review your notes, and which basically are the keys to taking all the notes. Are there specific mutations? What they would probably do, almost definitely, are going to tell you what the mutation does. It's not so much, and again, I would say that if it's anything, it's going to tell you specific. To understand every single mutation is not the key thing. I think the key thing is to understand, to reason out where this would occur based on data. So that's the answer. Your question. The question was, are there likely to be questions on specific mutations? But it'll be set up such that you don't necessarily have to do that nor waste time trying to look it up. But more importantly, again, the College Board has said that this year the questions are going to be such that you're just not going to be able to look up the answer per se, so you need to reason it out. So I would err on the side of being able to reason out given the information that they give you. And I'm going to try to get through this so that I could show you a good example. All right, so getting back to this, but thank you for the question. Prophase, you have to know your steps. Everything aligns in metaphase, and anaphase, things pull apart. And then telophase, you split into two. Interphase is your next phase. So have a way to memorize this as well as understand the basic things of each one of these, okay? And IPMAT is a good acronym in terms of that. So the bottom line is also what you come out of that. Daughter cells that are identical to the parent, and proper number of chromosomes, and that's the way the world works. Otherwise, we would be in all kinds of problems, right? Life would not exist if we didn't have that regulation. So sometimes you don't have to just pass through a membrane, but actually just deliver a message, and that's signal transduction. So that's basically a ligand. It's going to bind to a receptor on the outside, cause a change in the cell, and those are channels. Catalytic receptors, G protein links, and so forth. That's what was talked about when we talked about this in second video. It's just that is a nice introduction into the cell cycle. So that's why it's included here, to refresh your memory on where it sits in terms of the cell cycle. And we just mentioned G1, G2, and S phase. And you always have these checkpoints, which is important to note. Cancer, of course, is you're going to have abnormal growth plus metastasis or spread, in case you know that word. You're going to have tumor suppressor genes that are going to prevent and basically prevent the cell from dividing when it shouldn't. And that's the key thing, because that's the uncontrollable part of this thing. Proto-oncogenes are also going to help the cell divide and go from there. We talked about mitosis, which is your division, your PMAT. We talked about in terms of that, so that's a good review. All right, we're going to have to talk about a couple of things about heredity, just so that you have a good understanding of at least what the stuff that you would remember, need to remember, and so forth and so on. Now you're talking about, before we talk about heredity, let's talk about the meiosis part of it, right? That's your production of your gametes, Okay, that's in the gonads. You'll either have your testes, ovaries, okay, special germ cells that start out. It's either going to be haploid or it's going to be a diploid number in terms of that. Meiosis is a big plus when it comes to genetic variation, especially because that's the driving force for evolution. And again, they're not going to probably ask anything on the evolutionary side because that's more of the stuff that you would have gotten prior to the COVID scenario. Again, but just probably the basics to note there. Meiosis 1 in terms of the division, meiosis 2 and what happens in each one. So you have a diploid and a haploid. Okay, so in prophase 1, it's a little bit more complex because what you have is things lining up side to side, and they call that the synapsis in terms of that. So that's what you have there. And it's, again, just the basics of it to note and make sure that you know the sequencing of it. This synapsis involves a triad, okay? And this is the crossing over occur also. So, noting that this occurs is important, okay? You have a metaphase plate that occurs, okay, and anaphase 1. So each one of these, again, noting, and they're summarized in the end for you. So for the purpose of the meiotic division, to separate the cystic chromatids, right, and basically have something virtually identical to mitosis. So you have mitosis 2, prophase, PMAT2 for each one, and then the cytokinesis, and you're all set. This is pretty much a good summary as to everything. As long as you know this and you get this straight and you're able to produce this, you'll be set. Again, you won't be asked to reproduce this. These are the details that are useful that you could note maybe in a sentence or two as you work through a specific free response question that they're going to give you. Okay, so noting the differences between these two is very important. So I highlight this. Okay, mitotic errors can occur because of disjunction, so you're not going to separate appropriately. And again, I mentioned and alluded to, but you're either going to have a diploid or you're going to have a discussion of a haploid, so make sure you understand that. 46 is your diploid number for humans, 20, but there's only 23. To be asked a specific one, and the names of everything probably won't occur. They'll just, you'll be told what it is and what occurs, and again, go back to the basics in terms of cell cycle, cell division, and so forth. Most everything is diploid, and then you have the gametes that are the haploid. Okay, so with this, and because this is genetics, the fundamentals, you have traits, right? And these things are going to be influenced on the genes. And what a gene is, we already talked about, is just part of DNA that's going to code for something in particular, your blue eyes, your curly hair, and so forth. And that's going to be on a locus. Okay, when you have two sets of them, you usually have two copies of a gene, one from each of the homologs, if you will. Okay. And then the alleles are on the same. So if we're talking about the height of the pea plant, then there's an allele for tall and face. Okay. Okay. All right. So where am I? Hang on 1 second. Okay. So fundamentals, phenotype, obviously your physical, and genotype are your in terms of the genetic makeup of cells. Okay. And what's the question here? Prophase. Okay. It's considered past you're asking me about prophase. It follows the prophase, but it's precedes the metaphase. So you can say that, but it isn't. It's a separate step per se, if that's what you're asking me. But usually, if you know the P and the M, you're all set in terms of that. Okay. Dominant versus recessive, again, we can go through all that. If you have a question about, the other question is, are they going to have the labs? From my understanding, from what I gather, they can ask you a lab, but the labs have specific details that we're targeting that's still basis. So you could have a lab, for example, that talks about, let's see, you can have a lab that talks about DNA sequencing, or, for example, you may have had one on Hardy-Weinberg, which won't be tested on there. So the point is understanding the concepts, not necessarily that the fact that you did a lab and that you had to do it this way in terms of look at the microscope and do this kind of thing. It's the theory part which is included in what you're studying now, to answer your question. The question was, will questions include anything about labs that we did in school? Okay. Okay. So dominant versus recessive, and everything associated with this, the genotypes you need to know. You need to know, again, basically your parent and how to write out the symbols, meaning that just know, capital T, small t, whatever it is that you're given in the problem. For Mendelian genetics, you have the law of dominance to know how to do a basic Punnett square, okay? Law of segregation in terms of being able to look when you have heterozygotes involved and being able to do a simple Punnett square. Independent assortment when you're dealing with, in a case like you have two of these different traits, tall and green versus yellow, knowing how these combine together is also important. Doing a more expanded Punnett square is also of use. And the basic idea of probability, if you have the probability of two things that are independent, the probability of this and that, you would multiply the two, okay? And then there's mutual exclusivity in terms of that. So if you have a tall yellow plant, you simply multiply the two probabilities. You need to the probability of tall and the probability of yellow, and then multiply those two together. So that's the idea, and this is the part of the multiplication part. Okay. So these are the three, the Mendelian genetics in terms of that, being able to just mention it, understanding its applicability in a specific problem. And then, of course, the test cross in terms of doing this is also very important. Okay. Sometimes they're on the same chromosome, so they move as a group. And that's the idea of linked and the idea of a dihybrid. You have recombinants in terms of a frequency. Okay, so you can add up the recombinants and divide by the total number. So that's a percentage of how far apart in terms of map units you have. So this is an example of something like this in terms of two genes that are linked and what the percentages associated are. You have sex linked when you're dealing with 23 pairs of chromosomes, with color blindness and hemophilia being a very big ones as examples, okay? And in this kind of case, you can have a mother and a father crossed and find which ones are the colorblind. So knowing the X-linked patterns are also good. Things that are non-Mendelian, genetic-wise, you have blending, inheritance, codominance. Each one of these, you should have at least an understanding. It is possible you could see a pedigree and to note the basics, but it isn't difficult to read through, so you have to follow along with it, as well as any environmental effects. And this goes back to, I believe, a question before about environmental effects that affect different things. Height and weight in humans can be controlled by genetic and environmental factors, but other things involved, let's say an environment can affect the soil of something and its pH and accessibility, or seasonal fur in Arctic animals, or sex determination in reptiles. However, again, like I said, if you're talking about the singular cellular level, then there are other ways to look at this. So genetics is about hereditary. We talked about diploids, homozygous, make sure you have that straight. Physical expression of traits, know your idea of the homozygous versus the heterozygous. And obviously the allele that wins out is going to direct the phenotype. Okay. I just touched on the Mendelian points, but these are the key: the dominance, segregation, and independent assortment. And if two genes are on the same, they could be linked, of course, and they travel together during the gamete formation. 23 pairs are going to give you 46. 22 autosomes, you have the two that are the sex chromosomes. As you know, the two X's give you a female, and the Y is the male. Importance. Notice that one, you have a condensing of a bar body that just gets dissipated in terms of that. Non-Mendelian are incomplete codominance and polygenic inheritance. And a couple of other ideas that we talked about with meiosis 1 and 2. Now, the last thing I'll leave you with is the idea of the free response. Okay. The key things to free response would be to make sure that you understand your pacing, that the amount of time that you know you have, to think like the grader. In other words, make sure that you follow the rules, the directions, to make sure that you do what you're told to do and answer the question. Make sure you read the whole question a couple of times first so that you gather your thoughts. Think before you begin to write, and use the space, because you don't want to use too much space that you go overboard in terms of that, not just with that, but with the time factor. Clarity trumps everything. Of course, you have to be accurate in what you say, but be able to clearly explain in a clear reasoning pattern what you have, and very important. And I did allude to this in the first video in terms of common sense dictates as well as you deal with that. There are tasks that you're told. If you need to describe something, that means you got to provide characteristics. If you need to explain, you have to explain how or why, depending on the scenario. In order to identify, you need to indicate information. In order to justify, you get the claim, and then you provide the reasoning to explain how it works. And then finally, to predict, you describe what you have. Okay? You could be given in an example, and this is a long question, but the bottom line is it discusses homeostasis, okay? And it gives you a table of treated versus the presence of a protein X or protein Y and the mRNA levels. And in just a cursory level, once you're given that, it's going to ask you to do things. You won't get probably anything that asks you to construct any graph. They're going away from that probably, where you won't have to draw, but you're able to type your answers in. But you still would have to analyze and describe and predict whatever they may have. So they'll have it set up for you that you need to do different things. Again, probably not construct, but you can always describe what a graph is. And then analyze the data for specific expression, whatever the case might be, and further predict. Again, so you need to think through. Here's an example you would describe, and again, this is an example of a part of a question where they're just basically asking you, they mention homeostasis and they ask you to explain it. Okay, so you would get the point or two, whatever is associated for that. Now, while you wouldn't construct a graph, you would be given a graph per se, and then you would be able to elucidate all the information. You would want to make sure you look at the title, that you look at what the units are, that you notice what the segregated data are, to make sure, and to see you have the presence of one gene, presence of two. So what happens when you have this gene present against against another one that isn't? And you would then continue writing as to addressing what the question is. And this is just an example, and trying to address and answer the question that's being asked. And again, it's not I'm just trying to show you how you would be able to give a written explanation for what it is that you have based on what they're saying. You can have, and this, within this writing is where you would use your reasoning of your biological knowledge. For example, it's likely that X is a repressor of transcription. It could bind to the promoter or another transcriptional factor to prevent the RNA polymerase from binding. Again, it's clear thinking without the specifics, but knowing full well that it addresses the question. mRNA and protein levels did not change, which means the enzyme activity somehow enhanced. So again, you're linking, of course, what the data are and what your observations are. And then, they'll give you another part of it where they'll say, well, and in this case, suppose this occurred, and then you would have that set up. All right. Usually one part's going to reference to an earlier one. You treat it as its own question. It shouldn't be difficult to remember, but notice that you have your own space to write. And again, I don't think drawing is going to be part of that, this one here, in terms of that they're downplaying that so that you could do that. Here's another one that was taken from from a previous one, and I just wrote a specific idea here. And this is a table where they basically tell you a researcher is studying patterns for gene expression in mice. And basically the researcher collects samples from 6 different tissues in a healthy mouse and measures the amount of mRNA. The M stands for moderate, H stands for high, and if you see a blank box, you have nothing. So these are the data that you're given. So this is the expression levels of 6 genes, for example, labeled as such. So question is going to basically ask you, identify the gene that's most likely to encode a specific protein that's essential in the component of glycolysis, okay, and prove your reasoning methodology. So you would look at this and say, okay, well, if I look at each one of these, I want to make sure that I have this gene C is expressed in all 6 of these different organs, if you will, in terms of that. So if it's expressed in all of these, it's actually tissues, right? Where an organ's a group of that. So the correct terminology would be tissues. Because it's being expressed in something like this, then that would be the gene of interest, because glycolysis occurs in all of this. So now you have to remember your glycolysis idea. And then it also says, for example, that the researcher also observed that tissues with a high level of gene H, or in this case, I should say gene D, high levels of gene D did not always have the gene D protein. So why would that happen? So why would that happen in a case like this? And you could say a couple of different things. You could say that the mRNA was not exported from the nucleus. You could say that the mRNA was not translated, or there was some kind of interference in the RNA that prevented the translating of this, or there was some kind of post-transcriptional modifications. All right, so hopefully that gives you an idea of everything. I've run out of time. I apologize. I want to just say thank you very much for everything. And I wish you all the best. If you have any questions, also look at our website, www.princetonreview.com. It's been an absolute pleasure. I wish you the very best of luck, and hopefully this presentation was helpful. All right, take care of yourself.